Hi. So I just mentioned the hydrogen bonding in DNA. You know, DNA is an enormous molecule. It can be just, tremendous. But many large molecules have hydrogen bonding that happens within the molecules. And one of the most important examples of that is what happens with proteins. So I'm going to go to OneNote and show you a picture, a slide basically, of 3 typical proteins. So let me go ahead and do that. Share my screen. OneNote. Okay. So here is a slide. Right here. And what you see here are 3 different proteins. And they're labeled. This one is human, I can't pronounce it, apomyoglobin. This is hen egg white, and this is an immune system protein. And what I want to show you is the dark blue lines are the connections between the amino acids and the protein. That's the protein backbone. Now, these are very cartoonish looking pictures, especially the ones in the upper right corner of each picture, because they're basically showing, whether there's an alpha helix or beta sheets or things like that, these overall structures in the protein. But what I want to focus on is not so much these dark blue lines, which are the, like I said, the links between the different amino acids. What I want to show you is these gray lines. So here and here, look at all these gray lines and the green lines as well. All these gray and green lines in all of these different structures are hydrogen bonds. And you could see, for example, this one here is holding this juncture, this one together with that one. So what these are doing, what these hydrogen bonds do, is they help the protein fold in such a way that is needed for that protein to function properly and do its job in the cell. Okay, so that's very typical of proteins. Now, something else I would like to tell you is that if you have a protein in an aqueous environment, so that means you've got lots of waters floating around, okay? So when the protein folds, okay, you would want to have the more water-liking, we call those hydrophilic. That's water-loving. You would want to have the amino acids that have more water-loving groups, which basically means polar, okay, groups on the outside. And then the more hydrophobic means water-hating, or water-fearing, but water-hating groups are on the inside. That's generally the way it works in an aqueous environment. So if you were in a non-aqueous environment, like suppose in a lipid fat, like in a lipid bilayer, you would have the proteins would fold in a different way. They would fold so that the water-loving groups, the polar groups, are on the inside, okay? And the non-polar groups on the outside, which would interact favorably. They would be more attracted to the lipids, right? The non-polar environment, so that lipids are non-polar. Okay, so that's the case for proteins. Now, maybe you're interested in knowing, what happens when you boil an egg, for example. So an egg, that's egg white, right? That's albumin. That's a protein. Okay, so what happens when you boil an egg? So the egg, it starts out, it's, kind of clear colored, it goes from clear to white. Okay, basically what happens is that the heat provides energy, and that breaks up the hydrogen bonds. So it provides energy to disturb or break up those hydrogen bonds so they can no longer function in keeping the protein folded. Anymore. So what is the result? Is that the protein unfolds. And guess what? When it unfolds, all those nice water-loving groups, all those nice water-hating groups are now on the outside. They're no longer on the inside. So the hydrophobic groups are not hidden on the inside anymore. And so the protein drops out of solution. It's no longer dissolved. In aqueous environment. In other words, water. Okay, it's no longer soluble in water. And so you get this solid looking egg white that isn't going to redissolve no matter what you try. Okay. I wanted to also go through this silly slide. I don't really want to spend time in class on this, but I think it's a good thing to go over. Okay, so I guess I should call this video protein folding and silly video on what is in the gas phase above a sample of water. So here is a glass of water, magnified many, many times. So these are H2O molecules. Now, I've been talking about how you have gaseous H2O in the liquid above, you have gaseous H2O above a sample of liquid water. And that, we have a certain vapor pressure of water. So this diagram here shows H2O molecules. This is what actually exists in a sample above liquid water. This is correct. Here, it's speculating that maybe there's hydrogen atoms and oxygen atoms above a sample of water. Well, that's just not true. In order to break those water molecules apart and tear it apart into hydrogen atoms and oxygen atoms, you'd have to supply way more energy than what these molecules have in terms of their motion energy, so they could occasionally escape into the gas phase. You would need way more energy. It's nowhere near the energy that would be required to break up water molecules into these atoms. Okay, so you might ask, well, what about H2 molecules and O2 molecules? Are they up there in the gas phase above water? And the answer to that is no as well. When water goes from liquid to gas, the molecules do not break apart. That would require too much energy. Okay, so you are able to, the kinetic energy of molecules moving in water are able to, that kinetic energy supplies enough energy for molecules of water to break away from the liquid. In other words, break up the hydrogen bonds, but it will not be able to be enough energy to break that molecule apart in any way, shape, or form. Okay, so this is what is present above a glass of water. You have liquid H2O, and then you've got some gaseous molecules H2O above it. Of course, you still have air above it. That doesn't exclude the fact that you still have air. So air plus gaseous water molecules, but no H2, no O2, no HO atoms. That's just not present above a glass of water.